So our first speaker, let me say a little bit about the biography of the speakers and a little bit about their abstracts are all in the book. So when I introduce speakers, I'm not going to spend a lot of time on their credentials or past accomplishments. But our first speaker is typical of those individuals who has really thought through the problem of gene expression and the problem of silencing genes and the problem of the genome structure, right, and the use of large data sets to gain information and hypotheses. And that's Andrew Chess of Harvard University, who really has pioneered our understanding of epigenetics, our understanding of allele-specific gene expression, and the consequences of that in pathology and in normal situations. And Andy is going to talk to us about genome-wide allele-specific analysis. It's a pleasure to introduce Andy Chess.
Thank you for the introduction, Arnie, and thank you for the invitation. I'm delighted to be here. I actually spent a lot of joyous time in this room a number of years ago. I used to come down here for the concerts when Robert Taub was the artist in residence here in the 1990s, and he was going through the entire set of Beethoven piano sonatas. He was sitting right here with the Steinway piano and stuff. Anyway, so it's kind of fun for me to be here now on this stage. I never was on the stage when he was giving his concerts, but okay, good. Good. So I'm going to talk today about a number of projects that we've been doing at my lab at Mass General and in collaboration with people at the Broad Institute to look at a variety of different aspects of regulation of gene expression. I'm basically going to talk about two different stories, and both of them have potential to impact on understanding of development and where it goes awry in cancer. The first one has to do with allele-specific expression, and the second one has to do with DNA methylation. So the first story, this is work that was pioneered by Alexander Sasha Gimmelbrandt in my lab. He was a postdoc, and now he has his own faculty appointment at Harvard in the pathology department, and also with John Hutchinson and Benjamin Thompson. And Ben Thompson's kind of interesting. He was actually a neighbor of Sasha Gimmelbrandt, and his day job is that he's a rocket scientist. For real. He's like designs like systems that target stuff for one of the military contractors in the Boston area, but he's a physicist, and he was interested in some of the problems that they were talking about, I guess, over beers and stuff. And so he actually wound up becoming a member of the lab as a visiting scientist and doing a lot of the programming that allowed us to do some of the work that I'm going to present today. So when you think about gene expression, generally when a gene gets turned on, you assume that both the copy that you inherited from mother and father, the maternal and the paternal copies, that both of them get turned on. But there are instances in which there's monoallelic expression. And this slide just goes over the general outline of the kinds of genes that undergo monoallelic expression and what kinds of patterns they have. So first I want to talk just briefly and put off to the side imprinted genes. So these are genes where there's a deterministic choice. A given gene is expressed always from some genes from the maternal allele, and other genes are expressed always from the paternal allele. And this is in this diagram, I just show a green arrow coming off of the maternal allele, and the paternal allele is in red and is silence. So in these cases, there are marks that are placed on the genes during gametogenesis that allow for this different expression. So these are very interesting, and sometimes we wind up studying them a little bit, but mostly in my lab we've been focused on monoallelic expression, which is of the random type. So here depicted some cell, this is one cell and this is another cell, and the depiction of a gene is on from the maternal allele here and the paternal allele here. So you have some cells expressing one and some cells expressing the other. These two sequences can be identical, so it's a very interesting question. How do you turn on one gene one allele in one kind of cell and another allele in the other kind of cell when they're identical. So in terms of the random choice, of course, the most well-known example of this for decades has been X inactivation in female mammalian cells, where because females have 2 X's and males have 1, there's a mechanism that allows for the equalization of the amount of protein that's made from X-linked genes, which is X inactivation. The entire chromosome, either maternal or paternal chromosome in each cell, gets inactivated early in development, and then it's maintained in the daughter cells that arise from that. So on autosomes, it was known for a long time that back from around when I was born, that the immunoglobulins, the genes that encode antibodies, are expressed from only one of 2 alleles because of the process of DNA rearrangement that occurs. It was actually, this was known even before the genes were cloned and DNA rearrangement was found. And of course, T-cell receptors, the other major arm of the immune system, also uses DNA rearrangement and has monoallelic expression. So this is so-called allelic exclusion. And so then in the 1990s, I showed that the odorant receptor genes, the genes that encode the receptors for different smells, that they also have monoallelic expression that is also random. And so these are now autosomal genes that have this property. And this led to, we looked at some other genes, and a few other labs around the world looked at selected other genes, like interleukins and a few other genes that showed this property. And they tended to be things that were involved in the immune system or in chemosensory systems. So with the advent of the sequencing of the human genome and then arrays that were made to interrogate single nucleotide polymorphisms, differences between the alleles, and these were made for population genetics and for studies of complex human diseases, we were able to start looking at this issue at a genome-wide level, and that's what I'm going to present now. So this just shows the different kinds of monoallelic expression in a different format. So imprinted, of course, as I said before, it would be, for some genes, it might be always just one allele expressed. X inactivation, if you look at a population or a tissue, lots of cells together, you'll see biallelic expression, which I depict as a yellow circle here. Or in clones or individual cells, you see either the maternal or paternal allele expressed. And then on autosomes, we have some clones that express both alleles and others that express maternal and paternal, or the paternal allele. Okay, so we've studied mostly lymphoblasts and fibroblasts, but also some fresh tissues, which I'll show some of the data for also. So this is something that can be seen in culture and also in fresh tissue from people. And so the way that we did the experiment was if this is a depiction of a cell and expression coming from only the maternal allele of this particular gene in a nucleus, and then here are the transcripts in the cytoplasm. If you look at genomic DNA, and let's say that it's a heterozygous genotype, and the way Affymetrix, which the company that makes the arrays that we were using here, they call the genotypes the A allele or the B allele. So we're just using that nomenclature. So if this is a heterozygous genomic DNA, you would get a genotype of AB, and then in cDNA here, you'd get only the A allele showing up because the B allele, the paternal allele in this case, is silent. So you'd see if you genotype the cDNA, you get evidence for monoallelic expression. So when we first were thinking about doing these experiments, we were a little bit upset when we saw how few of the SNPs that are on the Affymetrix array. So at the time we were doing this, it was array sets that had, first they had 100,000, then they moved to 500,000 different SNPs. Now they're up to around 1 million SNPs and even more coming out soon. But most of them were not, only 1% of them were in the actual mature messenger RNA. So that was a big hit to take, a 99% loss. But then we realized that, well, if we can take advantage of the probes that are in introns, that we'd be able to do much better experiments. So realizing that in the nucleus, there's a lot of unspliced message and also introns that have been spliced but have not been degraded. You can actually convert that nuclear RNA into cDNA, and that's just depicted what we do here. And then we just put this into the genotyping platform. So basically, we take our material, our experiment, which is making cDNA from the nuclear RNA, we make it into a material that we can then give to a genome center, whether it's Eric Lander's Genome Center up in Boston, or we can FedEx them other places, and we send it off, and we tell them just to treat this as if this is a clinical sample of genomic DNA that someone's doing some study to look for genes involved in complex disease, and send us back the data. Now, of course, we tell them, don't worry that most of the genotypes are going to fail because lots of genes are not expressed in the tissue that we're sending. Just send us back the raw data, the actual intensities, what they call the cell file, which has the intensities, and we can actually extract everything that we need from that. So this is just an example showing that the intronic SNPs and the exonic ones, when they're in those rare cases where there is a SNP, a single nucleotide polymorphism in the mature message, that they agree with each other. So here is this last row here is a SNP in the coding region. These other three are in introns. And basically, these are the 4 different SNP IDs. And then these are the genotypes of the parents, of the uncloned DNA and cDNA. And so when there's a call made, it's always heterozygous. And then these are different clones made from this uncloned cell line. This is an EBV transformed B lymphoblast. So these H2, H3, all these different clones are different things. And so you can see that there are, in this case, this is a gene which is on the X chromosome. This is one of our control genes. So when we looked at the entire X chromosome, each row here represents a SNP along the X chromosome in order. And so you can see both all the big picture aspects of X inactivation, in that in a non-clonal cell line or a tissue you have biallelic expression, or in individual clones there's monoallelic expression. So all pink here, or mostly pink, or mostly blue, as the case may be. And then there are some areas where it's gold, and those are actually expected. This is some of the subtleties of X inactivation. In humans, it's known that there are genes that escape X inactivation, and these yellow bars that are showing up in our experiment correspond to those regions that had already been identified. So basically, we were able to see both the big picture and also subtle aspects of X inactivation using the study, using this kind of analysis. But this was just our control. So when we started assessing autosomes, I'll first show a couple of examples of genes. This is the gene called APP, or amyloid precursor protein, which has been implicated in Alzheimer's disease. And I'll just quickly go through how the SNP data that we get from the array allows us to see that this is monolithically expressed. So in this picture, if it's a white bar, it means that there was no expression detected. If it's a gray bar, expression was detected, but we can't tell which allele because the genomic DNA is homozygous. So there's no way to tell where the expression came from. But in the cases where it's colored, it means that there was expression detected and that the genotype was heterozygous. And you see either pink or blue. You see, if it's green, it means that we know it's monoallelic, but because the parental genotypes make it ambiguous for the child, which one is maternal and which is paternal, we know it's monoallelic, but we don't know which allele, whether it's maternal or paternal. So basically, if we then collapse down for each gene all the different SNPs into the ones that are informative, we can see a picture that looks like this. So here's different clones. This is biallelic. Here's monoallelic clone. This is actually the same clone after growing for an extra over a month in culture, showing the same kind of pattern. This is for one particular gene, but of course we did this for the entire genome. This is just some other examples of other genes that show this pattern of expression. And this is a fibroblast cell line here. So when we now look at the entire genome, we see that there are actually lots of genes on autosomes that have this property. This was something that was really unanticipated, given people's general assumption that X inactivation was, of course, an example of this, and there were a couple weird things like immunoglobulin genes and odorant receptors. But this shows that genes that are present all over the autosomal genome have this property. And this just shows the fraction that are monoallelic. And class 1 and class 2 just indicate how many SNPs and other aspects of how the data was analyzed, that how confident we are that it's monoallelic. So we're most confident in the class 1 ones. All of those verify when we use other kinds of experimental approaches. And around 60% or 70% of the ones which are class 2 also verify. And these are genes that are spread out. I'll show you a picture of their distribution across the genome in a moment, but I'll just tell you right now that they're all different kinds of genes, but there is an overrepresentation of genes for cell surface proteins. And so this leads to an interesting concept that perhaps one of the useful aspects of having this kind of random monoallelic expression is that it allows cells to become distinguished from one another. Even individual cells with the same expression pattern of all the same genes might wind up different because of different patterns of expression of the two alleles. And this just shows the patterns of across the genome. These are all the different human chromosomes, and you can see the distribution. They're spread out basically throughout the areas. These areas that don't have any are areas where there are no genes at all. And then chromosome 18 is shown here for a number of different clones, individual clones from different individuals. And what this slide shows is that along a given chromosome in a given clone, there is not a consistent tendency towards one or the other allele. So for example, we have paternal expression of a gene on this side of the chromosome and maternal expression of these genes here. So there's not a chromosome-wide coordination like one sees with the X inactivation. So it's some kind of random event that's happening early in development and maintained in the daughter cells at individual loci throughout the genome. So this is a control experiment, which is also important because it involved fresh tissue. And it's just depicted here a schematic of the experiment. We basically took small pieces of tissue from the placenta. And this is from placenta from a female individual. And so it's been known for a while that the placenta grows in clonal patches. So if you look at a given piece of tissue, it's likely, if you cut a small enough piece of tissue, it often has come from one single cell that then grew up into a piece of tissue. So we took small pieces of tissue and checked their X inactivation status. And those ones that were skewed, which was consistent with it being from one cell, we then analyzed those patches of tissue as if they were clonal cell lines like we had done in the earlier experiment. We saw a similar percentage of monoallelic expression in the placental as we did in pairs of clones from the clonal cell line. So what this shows is that the same kind of phenomenon is going on in this in vivo tissue. And we could also do another kind of experiment. This is also fresh tissue. This involves taking white blood cells taken from the peripheral blood and doing RNA fish, so we have probe in green for DNA and red for RNA. And this is a cell which is expressing from both alleles, and this is a cell which is expressing from one allele. The blue is just labeling all the DNA as a background. Okay, so one of the interesting things that we wanted to address was whether, even if the two genes are not different from one another, the two alleles are not different from one another, could there be a difference between cells mediated by the monoallelic versus biallelic expression. And so to look at that, we looked at the levels of expression in clones that are making one allele versus the level of expression in other clones that are making the gene from both alleles. And if there's feedback regulation of the gene, then you would expect those two to be equivalent, whereas if the number of alleles that are turned on or the number of pieces of DNA that are activated for that particular gene is churning out for each one the same amount of expression, then you'll have twice as much expression in those cells. So for APP, this just shows that biallelic clones in general, this is just from real-time quantitative PCR experiments, you can see that the clones that are biallelic tend to have around twice as much expression as clones that are monoallelic. So some genes show this kind of pattern. Other genes that we've analyzed show a pattern where it's roughly equal. So this shows that at least in certain cases, you can actually have a distinguishing mark for a given clonal cell that is determined by whether it's expressing one or two alleles of a particular gene. Okay, so what I've shown in this section is that there are perhaps more genes on autosomes subject to random monoallelic expression than we expected. It's maybe more than on the X chromosome, that they're distributed across the genome. And there are many different kinds of genes are affected, but that there is a preponderance of genes that are expressed on the cell surface. And then this can affect the level of expression. And so this then allows for a difference between cells, even if the two alleles don't have a code a different protein. And so we also, I'm not going to present the data in the interest of time, but we also now have similar data in the mouse, using mouse clonal cell lines, show the same kind of phenomenon. And genes that are involved are not entirely overlapping with the human genes, but are a significant amount of overlap, around half of them. So this just shows the potential for this kind of skewed X inactivation to impact on different clinical phenotypes. And so as an example, I show where this kind of thing has been clearly demonstrated for an X-linked, this is a pedigree for an X-linked disease called factor VIII deficiency. And so these are affected males. And here, where the arrow is an affected female. And so you wonder, well, if this is factor VIII is a soluble factor, and so females generally don't get the disease because if half their cells are making it, that makes enough of it to be circulating in the blood. And so usually it's just males affected. But this female was affected because she also, in addition to having one bad allele for the factor VIII gene, had skewed X inactivation for some other reason that led to most of the cells, or if not perhaps all of them, expressing only the mutated allele. So she was affected. So if similar kinds of skewed patterns of activation and inactivation of the alleles are occurring on autosomes, this could lead to differences in phenotype. And we have a number of projects in the lab ongoing right now to look at various different potential human diseases that might be impacted by this kind of expression. Okay, so some of the things that we're looking at now involve looking at things like monozygotic twins to see if this also can be an explanation for why they sometimes differ, looking at skewing of X inactivation and skewing of autosomal activation. And we're also interested in implications for cancer. And so in terms of cancer, one thing is that this could be a potential cause. One thing that I didn't mention is that, of course, on our list of genes that are showing this random monoallelic expression, tumor suppressor genes are not showing up, because it would make sense that would be a bad idea to have tumor suppressor genes subject to this kind of expression, because if you had only one allele available and that got mutated, then that would lead to cancer. So maybe that does happen sometimes, but we haven't seen those genes showing up in the lymphoblast clones yet. But it's certainly potential involvement as a cause. But the other thing that needs to be considered is as people start taking approaches to looking at cancer genomes and cancer expression across the genome and the epigenomic modifications that are occurring, a lot of them might be associated with this process, which is a normal process occurring in all cells of random monoallelic expression for a particular subset of genes, around 8% of genes. And so there might be differences between the two alleles which are not part of the cancer, but are just present because a single cell happened to be transformed that gave rise to the cancer. So this is something that definitely needs to be considered in terms of those kinds of issues. Okay, so now I'm going to switch gears and talk about DNA methylation. And this is work that was done by Asaf Hellman when he was a postdoc in my lab, and he's now at Hebrew University in Jerusalem on the faculty. And John Hutchinson and Yoshiko Mito are currently in my lab, and John and Yoshiko are doing aspects of the work that's ongoing right now. So when we wanted to start studying DNA methylation, we were also going to be taking a genome-wide approach to this. But we started by looking at the X chromosome, because in the textbooks there was very clear prediction of what we should observe when we started to look for methylation patterns. And that is, so X inactivation, as I mentioned before, you have in female cells, two chromosomes that can be similar or identical sequence in the case of mice, but they're regulated differently. And so we wanted to analyze those as a control, and then we went on to looking at the entire autosomal genome, looking at single nucleotide polymorphisms and methylation. So in terms of X inactivation, these are all aspects of it that I mentioned, that it occurs early in development and then it's maintained. So clonal cell lines are a good way to look at this. And what's known about X inactivation is a lot of different things about the epigenetic modifications, including a non-coding RNA called EXIST, which X inactivation specific transcript. There's hypoacetylation. There's different histone subunit usage. There's late replication, and there's also DNA methylation. And what was in the literature was the idea that the CPG islands, which are collections of high density of CPG dinucleotides, especially near promoters of genes, a few of them had been analyzed, and this led to the idea that there is aberrant methylation, not aberrant, but unusual methylation, because normally these CPG islands are unmethylated, in contrast to most CPGs in the entire genome. These are thought to be unmethylated mostly, but on the X inactivated chromosomes, there's supposed to be methylation here. So we started to look at this at the genome level. So here we're also using the same kind of Affymetrix arrays that are designed to look at SNPs. We're again hijacking them for a different purpose, to look at a different part of the epigenetics, which is DNA methylation. And so the way that, I'll just quickly show you how this is done. The way that the SNP arrays work is that you actually amplify up a reduced representation of the genome using first digestion with NSP, here I'm showing NSP1 and STI1. These are just different restriction enzymes that cut. And then you PCR amplify, shown by these primers here. These are primers that allow amplification of regions across the entire genome. So roughly around 20% of the genome gets amplified in the experiment. And then that material gets labeled and put onto the hybridized onto the array. So if we use methyl-sensitive restriction enzymes, or MSREs, so here is shown an area of one particular amplicon where here's the SNP, it's an A on the A allele and a T on the B allele. And here's a site which has methylation on this B allele but not on the A allele. And this little scissors that I drew here is cutting the DNA that's unmethylated, because these are enzymes that only cut the DNA if it's unmethylated, but leave it intact if it's methylated. So what you can see here is that if you run this DNA through the genotyping protocol after you've cut with the MSRE, and I should just mention, these MSREs it was a cocktail that we're using of enzymes that together hit around 40% of the CPGs in the genome. After the hybridization to the array, you would only see signal coming from the B allele, because this molecule can't be amplified by these primers that are giving the genome-wide amplification. So this is very powerful because it allows us to compare the genomic DNA genotype to the DNA genotype that we get after MSRE treatment as giving us a picture of DNA methylation. This is an example from older kinds of arrays where we have perfect match and mismatch probes. And mismatch probes generally are dark. Yellow means just that there's intensity of hybridization. And this is genomic DNA for one clone showing both alleles present. And then in two different clones, we can see that there's, in one of them, methylation on the B allele, and the other one methylation on the A allele. So we're seeing opposite patterns. And then we can also make an unmethylated copy of the genome using a polymerase that copies the entire genome in a relatively uniform fashion, and then run that on the array just to make sure that there isn't a polymorphism in the restriction site that's allowing the cutting to occur. So when we started looking at this, we first, again, looked at the X chromosome, and we were pleased to see that there was monoallelic methylation. Roughly 1/3 of the SNPs in any given clone showed this. And so I say a switch to a hemizygous call because basically we know the machine is, if we use the machine software, it's actually saying that it's homozygous. It thinks that only one allele is there, but we know that that's really coming from only one of the two possible copies, so it's really one copy. And what we saw, we were surprised by that. There was actually more methylation on the active X than on the inactive X. And again, so these are SNPs that are not just in CPG islands near promoters, but they're spread out all throughout the X chromosome. And so this just shows the extent to which there is this kind of skewing. So the the extent to which each of these circles is filled in shows the amount, the fraction of the pie that represents the amount of X active specific methylation versus X inactive specific methylation. And so the average increase is around 2.5-fold in different experiments. This is just a control experiment showing that the normal methylation of the CPG islands near promoters on the inactive chromosome is intact. And this is just an assay that's commonly used around the world to assess that. So we're very interested in where do these methylated areas fall with respect to genes. Is it completely random across the X chromosome, or is it more concentrated in genes? And so by looking at all the different SNPs, we saw that almost half of them were in known genes, annotated transcripts, whereas only a quarter of them would be expected, or 27.5% would be expected to be in genes by chance. And so the p-value associated with this is indicated here. So it suggests that these modifications are present in intergenic areas and within genes, but they're more common within genes than would be expected if these were randomly distributed. And it's interesting to note that lots of the genes on the X chromosome are not actually expressed in the cell type that we're looking at. So this preponderance towards gene bodies of the methylation that's on the active X is something that is not just a consequence of transcription. It's something about the potential for transcription that somehow is known by these cells as they're accomplishing their DNA methylation patterns. So the old idea of CPG islands being methylated is still there, but now we add to that on gene bodies, or the entire transcribed area of the gene, here are the different exons and the introns, because these X active specific methylation, some of them fall within exons, but most of them are within introns, because the introns make up the bulk of the transcribed region. But if we look at the entire gene body, the transcribed regions, we see more methylation on the active X chromosome than inactive X. Okay, so now I'm going to present some of the analyses that we've been doing of SNPs across the entire genome and methylation that's nearby to them. So there are a lot of ways that are well known that single nucleotide polymorphism, SNPs, or other kinds of genetic changes can affect function. Of course, if it's non-synonymous, if it changes the protein coding region, then that can lead to a different function of the protein. Transcription factor binding sites can be affected. Splicing sites can be impacted by SNPs. And this can lead to differences in regulation of splicing or in transcription factor binding, respectively. And then, of course, there are other ways that are known also. For example, a couple of years ago, it was discovered that there were a large number of microRNAs in mammalian genomes. And so soon after that, people started looking at SNPs, and they saw SNPs in untranslated regions of genes often impacted the binding site for microRNAs. So you could have an impact on phenotype based on that kind of a mechanism. So we were interested in looking at the idea that SNPs might, and other DNA modifications might influence methylation of DNA in cis locally next to where the sequence difference was. And so one way this might occur, and this would be relatively easy to find, would be if there's an absolute difference. If always one allele is, and here fill in just means methylated and not filled in. So if in different individuals for this particular gene, it was always methylated on copies of the allele that were the A copy and never on the B copy, this would be an absolute difference. But if there's a difference in propensity, what it would mean is that there's a difference in the chance that one allele might be methylated or not, but not an absolute thing. Then you might see a picture like this, where, so here the A allele is clearly more methylated than the B allele, but that there are certainly individuals where there's methylation on both alleles or neither allele. And you might even have instances where if it was a heterozygous individual, that they might be methylated on the B allele and not the A allele. But overall, when analyzing a number of individuals, you would see the pattern emerging that there was a difference in propensity. So this is, of course, easiest to observe in heterozygotes, given the assay that we're doing, but we can also look at the raw intensity data and look at homozygous positions. And so again, we're using the same kind of an assay where the cutting with the MSRE cocktail leads to this A allele not being represented in the material that gets hybridized to the array. So we'd have an AB, or heterozygous genotype, switching to a BB genotyping, or B0, as we know it to be. And so this just shows the percentage of monoallelic methylation for one individual that we analyzed, looking at all the different chromosomes. And you can see that there's a fair fraction of amplicons. So each SNP is present on one of these amplicons that gets PCR amplified. And we see a fair fraction of them switching to one allele from having the heterozygous genotype, indicating monoallelic methylation. And to analyze whether there's some SNPs are more likely to be impacting this than other SNPs, we started looking at individuals. And so here I'll present data of comparison of a parent and a child. We also analyzed unrelated individuals, because a lot of this is being driven by common sequence variants that are present in the entire population. But here's a parent and child, and what you can see is if you look at SNPs which are heterozygous in both the father and the daughter, that so here's 12,000 SNPs. A lot of them stay as an AB genotype, indicating that both alleles are methylated. Some of them switch to no-call, which means that either they're both unmethylated or somehow in an intermediate state. But there is switching to, for both individuals, to a state that indicates monoallelic methylation. And if we then compare the number of SNPs which have the same epigenotype, in other words, both the father and child switching to AA, or both the father and child switching to BB, and compare that to the number of times where it's the opposite, where one individual switches to AA and the other switches to BB, those, if this was a completely random process, those would be expected to be equal to each other, and they're not. So we have a very clear bias towards the switching to the same epigenotype, and the p-value is indicated here for how much this differs from what would be expected if it was a 50-50 split. You'd expect around 230 for each category. Okay, so this just shows that it's coming also from the mother. The same kind of experiment can be done. So I just happened to show a father-to-child transmission that can come from either parent, and it also can be from unrelated individuals. So one of the things we wanted to make sure, since some DNA methylation patterns are known to be imprinted, in other words, be based on marks placed by during gametogenesis, either in sperm formation or egg formation, we wanted to rule that out. And so this was looking at 3 generations, and basically what we would expect if it was imprinting was playing a big role, we'd expect to see the same parent of origin in both generations more often than a switching of the parent of origin, and we see that they're roughly the same. So imprinting is not playing a major role here. Of course, it can do control by sulfite sequencing and also other kinds of controls involving PCR analyses to show that the array data are robust. And of course, we can find known imprinted differentially methylated regions, DMRs, and of course the X inactivation studies. These are all different things that are controls on the experiment. By sulfite sequencing, I just show here a table showing that these are some of the SNPs that we analyzed and the primers, and then the number of reads, and the inconsistencies with the array results are here, and they're all zeros because they were never inconsistent. It was always confirming what was on the array. Okay, so as I mentioned earlier, we've analyzed unrelated individuals, and we can see the same kind of thing. And this suggests that these SNPs influence to local methylation patterns is something that's present in these common variants that are present in the entire population. And we can also analyze different tissues. So the analyses that I presented thus far are based on lymphoblast cell lines, but we also have looked at whole blood and a variety of other fresh tissues that we got from a tissue bank, things like kidney or liver or brain. So you can look in different tissues. So this is likely something that's occurring early in development that's then present in lots of different tissues, or indeed all the tissues across the body. So analysis of siblings is particularly useful in this kind of experiment because they can, there are certain parts of the genome where any two siblings will share both of their copies of DNA identical by descent. So two alleles identical by descent means that they got the same copy from the mother gave the same of her two copies to each of the children, and the father also gave of his two copies the same one to each of the two children. But there's also areas of the genome where they share nothing identical by descent, and they're as unrelated as any two people within their general population. And then, of course, there are intermediate states where they share one allele identical by descent. So if we see greater similarity in the methylation patterns in the share 2 regions, this would be informative. So we looked at that issue, and this just shows in table form, and you can see different pairs of siblings in a family, and you can see that the amount of methylation that's present in a monoallelic pattern that is more than would be expected by just a chance occurrence is actually greater as you get into the 2 chromosome shared regions, and it's shown here in a graph form, which is more easy to see. And so here are the siblings with two. The pointer is dead, but two chromosomes identical by descent has the highest amount of overlap of allele-specific methylation. So the thing that's important to note here is that the data are a little bit noisy, and it's not, the allele-specific methylation is not observed in every individual, so you have to look at the picture across the entire genome in order to start seeing the patterns that are emerging. Okay, so another way of analyzing this is to look at whether, if the sequence has the SNP and the surrounding area is influencing methylation, you might expect it to sometimes to show a much closer distance than would be expected by chance to the MSRE site. So we can measure the distance from the MSRE site to the SNP along the amplicon of a given range. And then we can model that with simulations that would randomly place the SNP and the methylation site on equally sized fragments. And so what you can see here is that if you look at all of the different kinds of SNPs, there is no such propensity towards a closer distance between the MSRE and the SNP. But if you look at the ones where one of the two alleles is a CPG, so the CPG dinucleotide is well known to be a very mutable base because, and so it's often depleted seen in mammalian genomes. So lots of SNPs have one allele with a CPG and another without a CPG. So these are SNPs that are not impacting a cut site for the enzymes we're using, but they're just a SNP, but that one of them happens to be part of a CPG and the other one isn't. And if we focus only on those ones, CPGs with methylation overlap, what we can see is that here have just been the different distances from the MSRE to the SNP. You can see that there are more than expected by chance in the very close in. So it suggests that for at least that class of SNPs that there might be a very local effect. And that's interesting in the context of what's understood about the structure of DNA methyltransferases, the enzymes that add methylation. Some of them are shown to have cooperative behavior close by. Okay, this just shows a pedigree of a family and showing some of the SNPs that have a propensity for methylation of one allele but not the other. So here's a bunch of siblings, and you can see most of them have methylation on the A allele but not the B, but this sibling has methylation on both alleles, and these other families have similar kinds of propensity towards one or the other allele. And I mentioned that we've looked in a variety of different tissues, and I've mentioned that these are present throughout the genome, these kinds of differences. So in this next slide, I'm showing simulations of the entire genome with the idea of trying to get an idea for what fraction of the SNPs across the genome are actually impacted by this kind of phenomenon. So in any given individual comparison of two different individuals, we see a few percent difference in the SNPs that are methylated in one individual but not the other, more than is expected by chance. But that doesn't give you an idea of what's going on genome-wide because it can be different ones in different pairs of individuals. So to look at this, what we did was we simulated under different models the extent of methylation propensity difference between the two alleles for those SNPs that have this property, and then the fraction of SNPs across the genome that have this methylation propensity difference. And then we monitored a number of different parameters, including the methylation overlap between individuals and groups of 3 and 4 individuals, and also the opposite overlap. And the numbers that are lowest here, this is just a sum of the squares of the fractional differences. It shows that the low numbers show that these are the most likely models for what's going on. So it shows that around 10% of SNPs across the genome have around a 50% difference in methylation propensity associated with them. And so this is just a first attempt to give a genome-wide picture of what's going on here. And this will, of course, be fleshed out better once we've analyzed more and more individuals. If we take one of the models one of the simulations is giving the lowest score, one of the best simulations. You can see that it approximates the data very well for overlap amongst groups of 2, 3, and 4 individuals. This is the percentage of methylation overlap observed in these different groups. And of course, the expected data, if it was completely random, would be dropping down to 0 almost immediately as you added another individual in, a third and fourth individual. So what this shows is just the extent to which these are not absolute differences in methylation. It's not like for a given SNP that we're reporting on that the A allele in all individuals is going to be methylated and the B never methylated, but there's a difference in propensity which can emerge from analyses of the entire genome. So examination of the epigenome is of course a way to find genes that are involved in complex human diseases, and also since methylation is well known to play a role in turning on and off genes, and that's a major important aspect of cancer development, the propensity to have a pre-methylated or pre-unmethylated portion of the genome at particular genes that might be important for cancer might lead to differences in proliferation, which might be important for early stages of cancer development. And so that's one of the aspects that we're very interested in pursuing. And then, of course, we're right now in the lab developing new ways of looking at this, because the arrays that we're using in the studies I presented are, of course, extremely powerful, but with the new sequencing technologies that are available, at this point we kind of feel like the sky is the limit in terms of what kinds of things we can do. And so we're actually developing ways of looking at allele-specific methylation across the entire genome using sequencing technologies. And I mentioned the people who did the work while I was talking, and so I'll thank you all and happy to take questions. Yes.
A couple of questions, just hopefully quick. One, could this skewing be partially explained just by fitness as you go through the genesis? And then question two, with the apparent lack of control of gene usage, could this be a prognostic indicator for things with the example you showed, like Alzheimer's or some other diseases, that we could actually jump right on? Perhaps explaining why some people would don't extrapolate, some people do, and blah, blah, blah.
Yeah. So let me answer the second question first because I remember it. This idea about Alzheimer's disease is one that we're actually looking at specifically in the lab right now. So the key first experiment, of course, is to show that in the brain there is monoallelic expression, and that's a difficult experiment to do, but we're doing it. We're doing laser capture of single neurons from brain tissue from right now normal individuals, but later we'll do it from patients, and we're demonstrating, or we're asking whether it's monoallelic or biallelic expression. If it's monoallelic, then we can start looking in different areas where there's different amounts of pathology. Because the clear implication, I didn't mention this, but for the APP gene, there are reports that SNPs that impact on transcription factor binding actually associate with increased risk of disease. So there are other things pointing at the amount of APP made. And so if we have a situation where some individuals happen to have more skewing towards biallelic expression versus monoallelic, or maybe the two alleles are slightly different from one another, that could certainly impact on disease. Now, the first question was about something about fitness and during development. Yeah. So anytime. So if I understand correctly, it's talking about the idea that you can have fitness of individual cells clones, right? So this is something that's been appreciated for a long time in terms of X inactivation, that you have the primary events which lead to the number of cells expressing maternal and paternal, but then afterwards you have sometimes differential cell growth. So even if you have equal numbers of the two alleles expressing early on, if one of them gives a growth advantage or growth disadvantage, you'll wind up with an imbalance after a while. That certainly could be playing a role in the autosomal genes, and we're looking into that in different kinds of clonal cell lines and also in mice in actual patches of tissue to where we can actually, and also in mice, you can do experiments where you knock in GFP into one allele and another color into the other allele and start looking at this. So we're starting to look at those kinds of issues now, and it's a great question.
We have one more.
Sure.
I'll make this thing on.
It's okay. Okay.
Speaking to it, no better.
Okay. So having both inherited monallelic expression and methylation would imply that there's a set of genes in our genome that aren't subject to sexual inheritance, where it's so has anybody looked at whether or not you get the sort of evolutionary differences between certain sets of genes that might or might not have these properties?
Right. So for genes that are subject to parent of origin imprinting, people have looked and they see what looks like evidence for conflict between males and females during evolution, leading to, for example, like classic examples, IGF-2 gene, which makes the embryos larger, is expressed from the male, the paternal copy, but not the maternal copy. And the IGF-2 receptor, which neutralizes that, is expressed from the maternal. But in terms of the genes that are randomly monolithically expressed, so there you wouldn't necessarily see any kind of issue with respect to which parent it came from. You have an individual cell's choice of one of the two alleles, but it's roughly half the cells making one, half the other. So from a whole organism level, you're not necessarily going to see something that would then lead to something that you would see as a sexual conflict or anything like that.
So let me just finish up by asking a question. It seems the emphasis you put on the genes which were inactivated allelically on a random basis results in preferentially inactivating alleles for proteins that are at the cell surface, as you said. And so that makes a tissue which we have often looked at as a homogeneous histologically and pathological tissue be composed of unique cells where in the combinatorics of all this, literally every cell in a particular tissue then becomes uniquely identifiable by its surface signature.
Right.
And so that I can understand as a mechanism to really have some individuality to cells. It's quite an interesting mechanism.
That was one of the things that we were very interested in. And right before we started on this work, we had been studying another mechanism which can lead to individual differences between cells that are otherwise identical, which was based on alternative splicing. That was in a completely different system. It was in the fly. We were studying this one gene called DSCAM, which stands for Down syndrome cell adhesion molecule, but it doesn't have anything to do with Down syndrome, but in the fly it has 38,000 different splice forms coming from one gene, right? More splice forms than the number of genes in a fly or human genome. And that's clearly, we showed there that that's clearly involved in the unique identity of individual neurons, and that's important for their wiring. So in the case of this random monolithic expression in autosomal, and that gene I should mention in humans and mice does not have alternative splicing, and so we were looking for some other mechanism that would give our neurons individuality, because we like to think of the mammalian brain as being at least as complex as the fly brain. So we thought of this as one possibility once we were getting this kind of data, that this might actually lead to differences. Of course, if the two alleles are encoding different proteins, then it's obvious you can get differences. But you can also get differences even with identical protein coding sequences, just based on the fact that some cells are expressing twice as much of certain products and and other cells are expressing half as much. So that definitely is a possibility. The other thing that we looked into for a while was the possibility that this could lead, we're always looking for things that make cancer cells unique. So if you actually know the particular pattern, and now you know the SNPs in all the different genes, which of course we'll know quite soon because all the sequencing is going on, you would be able to say, okay, this particular clone of cells, even if, which made the cancer, even if these genes, their monoallelic expression has nothing to do with the tumor genesis, it is a marker of that cell type. And so if you had something that somehow targeted that could be extremely powerful. Now, of course, the problem is that you talk to protein chemists about the idea of having a small molecule that targets one allele of based on a single amino acid difference. That's often very daunting, to say the least. Yeah.
Thank you very much. Thank you.